All right, this lecture is on the molecular basis of inheritance. In this chapter, we're going to go over some of the experiments that were conducted to determine whether the hereditary material was DNA or proteins. So this is going to be a search. The search for the genetic material. Now, scientists had determined that carbohydrates did not have enough variation. Those were put together by monosaccharides. And that lipids not enough variation either because those were glycerol and fatty acids. So the race was on to determine whether the genetic material was composed of proteins or DNA. Now proteins in the nucleus, the chromatin was 60% protein. We're going to find out later that that's because there's a lot of histone proteins as well as some proteins that are actually inside of the nucleus. It's very complex. There are 20 amino acids. So it had a 20-letter alphabet for coding for hereditary information, such as from bacteria to whales, grass, crickets, horses, humans. So it had a pretty complex 20-letter alphabet. So there was enough variation to code for protein production, but scientists just can't say that, they have to prove it. DNA, on the other hand, of the chromatin, DNA represented 35% of the chromatin content. And DNA was pretty simple. It only had 4 nucleotides, a 4-letter alphabet to code for the hereditary material. And they thought that that was not enough variation. But again, they have to prove it. Well, one of the experiments that was conducted was by a man named Frederick Griffith. He'd actually discovered bacterial transformation, and his work started in 1928. And what he did was he injected mice with a live S form of pneumococcus, and they developed pneumonia. So the S strain developed pneumonia. But when he injected live R bacteria, they did not develop pneumonia. Now the S is called S because of the way it grows on a bacterial plate, an agar plate. And you see that the border is nice and smooth. So the smooth colonies are called the S type, and they actually have a capsule around them that masks the pneumococcus from your immune system. The R type also grew on the agar plates, but it had a rough, jagged border, and so they called it the R type. Now it did not have a capsule around it, and so the glycoproteins were seen by the immune system, and the white blood cells could kill these bacteria. What Griffith actually discovered, it was discovered later on, was that bacteria can absorb DNA from the environment. He actually discovered transformation. Here we have the same information here about the mice, and I'm showing you the species A versus species B. And you can see the species A has a capsule around it which masks the bacteria from being seen by your white blood cells, whereas the R type on the right does not have that capsule, and it can be readily seen by your immune system and killed. So the capsule masks the bacteria from your immune system. Well, Griffith has mice injected with heat-killed S bacteria, did not develop pneumonia. Now they didn't develop pneumonia because the heat denatured all the enzymes. And so the enzymes are proteins. And so the enzymes no longer worked. Mice injected with the heat-killed S, which were dead, the proteins were denatured, and live R bacteria, some actually developed pneumonia. The R type, which were non-pathogenic, had been converted into the S-type bacteria, which would cause pneumonia. DNA was later discovered to be the transforming agent in 1944. Bacteria can absorb DNA from the environment, and the R bacteria had absorbed DNA from the heat-killed S bacteria. Some of them had popped and their DNA was released into the environment, and it was absorbed into the R bacteria and gave them the ability to produce a capsule. They had been transformed. Now, Hershey and Chase, since the race was on to determine whether it was proteins or DNA, which was the hereditary material, they found viruses, which are, remember, they are not living, but they used viruses because viruses have a protein coat, and inside these viruses they used had nucleic acid, DNA. And what they did is they labeled the T2 phage. Phages are bacteria which will infect viruses which will infect bacteria. So they labeled T2 phage protein, the outside protein coat, with an isotope of sulfur. And they labeled the T2 phage DNA with an isotope of phosphorus. Now you see it says a virus phage eater is called a bacteriophage. They attack bacteria. Which of the two, protein or DNA, was entering the cell to change the activity of those bacteria and turn them into virus factories? One of them was responsible, and the race was on to find out which one. Now what Hershey and Chase did is basically in one room, they got these bacteriophages and they put them in a medium that had an isotope of sulfur in it and bacteria. The virus infected the bacteria. The bacteria were turned into virus factories, and they made, you see at the upper right, new viruses, and it says the isotope of sulfur was incorporated into the protein capsule on the outside, the shell of this virus. In another room, they had some bacteriophages they put in a bacterial culture that had an isotope of phosphorus in it. Well, the virus turned the bacteria in that medium into virus factories, and they incorporated that phosphorus into the DNA. So you see the lower right picture, it says the isotope of phosphorus was incorporated into the viral DNA. So they were going to find out when the bacteriophage attacks the bacteria, what did it do? Did it somehow put its protein inside, or did it put its DNA inside to change the activity of that bacterium? The protein and DNA labeled phages were allowed to infect separate non-radioactive colonies. So they weren't put together. They were allowed to infect bacterial colonies in different plates. Like it was actually in a liquid medium. The cultures were agitated to shake the viruses off the bacteria. The mixtures were centrifuged, forcing the bacteria, since they're a lot larger and more dense, to the bottom, and the lighter viruses remained above in the liquid. When they analyzed the bacteria, they found that the radioactive isotope of phosphorus was found inside the bacteria, while the viral protein with the isotope of sulfur was outside the cells. Thus, DNA entered the cells and was the genetic material. The viral DNA was what was causing the bacteria to turn into virus factories, because the DNA that was labeled was found inside the bacteria. None of the protein was inside, just the DNA. Here's a picture showing just a little tube here. The virus protein coats were up in the medium up here at the top after the tube was centrifuged, and the bacteria containing the isotope of phosphorus were down in the lower layer. So the bacteria have the isotope in them of phosphorus, not sulfur. So that was, a check mark for DNA being the hereditary material. Okay, we have another experiment here. This guy, his name was Erwin CHARGAFF. In CHARGAFF'S rule, here's what he found out: determined that the amounts of adenine and thymine were equal. Well, that should be true because adenine base pairs with thymine, and that the amounts of cytosine and guanine were also equal, which that's true too because cytosine base pairs with guanine. So this should be 1-to-1 on both of those. He analyzed the human cells and found the AT and the GC ratios for humans. And they found out that those were different for cats, different for horses, different for certain fish. So different species have different AT and GC ratios. He also determined that the amounts of AT and GC were consistent within a species. So humans have the approximate AT-GC ratios just for humans. Other organisms have different AT-GC ratios. Those are the bases that make up their DNA. Some other evidence for DNA being the hereditary material. The amount of DNA remained constant within the daughter nuclei after mitosis. That means that the daughter cells had the exact same amount of DNA as the original parent cell, which we know that's true now. The DNA content of haploid cells, the gametes, was half that of the diploid cells. That's true too. Remember, gametes are haploid. They have 23 chromosomes. And diploid cells, the somatic cells of the body, have 46 chromosomes. And the amount of DNA was the same for all cells within a species except for gametes, which had half as much DNA. Okay. The double helix. Rosalind Franklin was an X-ray, and even Maurice Wilkins were X-ray crystallographers. It says X-ray crystallography of DNA. What they did is they subjected molecules to X-rays, and they would have a diffraction pattern on a plate, and by being able to read the X-ray crystallography plate, they could determine the shape of a molecule, the actual shape of the molecule. But they didn't analyze the picture. They showed it at a seminar, and James Watson and Francis Crick happened to be there, and they realized, Crick analyzed Franklin's picture. He realized something about that picture, and it would show he could see the shape of that molecule. And so Watson and Crick asked Wilkins for that X-ray crystallography picture, which was Rosalind Franklin's, and they analyzed it and found out that it was 2 nanometers wide. That's from side to side. That's 1 purine and 1 pyrimidine. It's either AT or GC. The bases were 0.3 nanometers long. That's actually tall. And 3.4 nanometers distance for the double-stranded DNA helix to make 1 full turn. Thus, 10 nucleotides are stacked in each turn. So they discovered that it was an alpha helix. It was spiraling, 2 strands that spiraled, and they figured out all the dimensions of this. They went on to get a Nobel Prize, and you don't hear about Wilkins, but you hear about Watson and Crick. And later on, Rosalind Franklin was exposed as the person who had the actual X-ray crystallography. The reason that Watson, Crick, and Wilkins actually got the Nobel Prize and Franklin didn't was that she had passed away from overexposure to X-rays, died of cancer. Wilkins, he isn't really one that we even talk about when we talk about DNA discovering either. It's always going to be Watson and Crick that you're going to read about. If I put it on a test, it's Watson and Crick. Molecular genetics. Okay, DNA structure. So a little bit about DNA. You remember the DNA, the building blocks of DNA are called nucleotides. So nucleotides are the building blocks, and they have 3 parts. A 5-carbon sugar, which is deoxyribose, a nitrogen base, one of those 4, adenine, guanine, thymine, or cytosine, and a phosphate. Those 3 things make up a nucleotide. And the nucleotides are linked into polymers, chains. And you know that DNA has 2 of these chains. It's double-stranded. So DNA is composed of a double strand of DNA polymers joined together by hydrogen bonds between their paired nitrogen bases. So far, what holds the adenine to the thymine is hydrogen bonds, and guanine to cytosine, hydrogen bonds. Here are 2 nucleotides, one that is responsible for making DNA on the left, and the other one on the right is for making RNA. You see that the only difference in the sugars, if you look at the deoxyribose sugar on the left and you look at the ribose sugar on the right, the only difference is at carbon number 2. You start counting from the oxygen, and you go clockwise, carbon 1, carbon 2 on the ribose, and you look at that and you'll see there's a hydroxyl hanging off of there. We'll look at deoxyribose, carbon 1, carbon 2, it just has a hydrogen. That's why it's called deoxy. It's missing the oxygen. Deoxyribose. So they have different sugars. DNA, look at the base. It has A, G, C, or T. Now, both RNA and DNA both have adenine, guanine, and cytosine. But DNA has thymine that pairs with adenine, where RNA has uracil that base pairs with adenine. So they have a different base, and they both have a phosphate. So you see there are similarities and dissimilarities, and that's all it takes for one to be the genetic material and the other one to be involved in several other things like protein synthesis is one. Here is one of the two strands of DNA. And if you look at the bottom here, you see that there's a sugar deoxyribose, that little blue pentagonal shape there. You have a phosphate, the PO4, and you have a nitrogen base, which is gray. That's one nucleotide. And you see that there's 2, 3, there's 4 nucleotides here. I was telling you something just a little bit ahead of time, is that if you look at the bottom of this strand, you see the sugar deoxyribose. Count over to carbon number 3. Starting at the oxygen, carbon 1, 2, 3, there's a hydroxyl there. Well, typically they'll pull that hydrogen off. That's called the 3' carbon, and that's an important carbon. Now keep counting over. Carbon 4, carbon 5's in the air, and there's a phosphate on carbon 5. Now that phosphate has an open bond that's going to bond to the carbon 3 of the next nucleotide. You see that? And that's how they bond. When they're put together, though, it goes 5' to 3', 5' to 3', 5' to 3'. So on this strand here, the bottom, where it says sugar deoxyribose, that carbon number 3 that has a hydroxyl on it, that hydrogen will actually be off. That'll be the 3' end. Look at the top of the strand. There's that phosphate. Look at that oxygen with the negative mark there. That's the 5' end of the molecule. So there's always going to be a 5' end and a 3' end of the strands of DNA. Now when they're put together, like if the top had the other strand that is a complementary strand to the right, the one on the left, that would be 5' at that phosphate, and the other strand would be 3'. So they're not lined up 5', 5', 3', 3'. It's 5' on one strand, 3' on the other. Here is one way of drawing on the left. You see all these nucleotides are just stacked. There's 4 of them on the left, and there's 4 of them on the right. And you see that it goes sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate on both sides. And the bases are base pairing across, and those dotted lines represent the hydrogen bonds between the bases. You can see adenine pairs with thymine, guanine pairs with cytosine, thymine pairs with adenine, cytosine compares with guanine across the middle. Now also look at the two strands. Look at the one on the left. You see that hydroxyl at the very bottom? I told you that hydrogen will be off of there. That'll be the 3' end. Now look at the other end of that strand. There's the phosphate, and that hydrogen will be gone too. That's the 5' end. Well, look to the right. This one's upside down, and there's that hydroxyl, which that hydrogen will be gone. That's the 3' end. And look at the bottom. That hydrogen will be gone off the phosphate, and that'll be the 5' end. So see, it's how they're arranged: 5', 3', 3', 5'. Now the picture on the right, these little ribbons represent the alternating sugar phosphates, because it's still the same sugar, phosphate, sugar, phosphate, sugar, phosphate. So that's not really that important to look at. What the important thing is the base sequence. The base sequence is what stores the hereditary material. And so they show the bases, and the little dotted lines represent hydrogen bonds from one side to the other. Now this is DNA replication, and this is how it actually happens. We'll look at that in just a moment. This is called the semi-conservative method here. In DNA replication, you can see that the strands are going to separate. So in the writing over here, it says the two strands separate, and each strand serves as a template to reconstruct the other strand. So the original strand's blue, and the reconstruction of the missing strand is yellow. An enzyme unzips the two strands of DNA, which we'll look at that in just a few slides. But you see this when DNA is copied, the two, which we have called sister chromatids, have one original strand and one new strand for each one, and they're held together by the kinetochore, right? People had to present different methods that they thought DNA could use to copy itself. And this one, first one, is called the conservative method. And basically they said that the DNA double helix would just copy itself like a Xerox machine and just peel off another strand, a brand new strand of DNA. Well, that one's not how it happens. This one's the dispersive method. This one is really weird, but somebody had to, propose it. And what it says is that the DNA double helix fragments and then fills in the missing pieces between the fragments to make up the two new strands. So DNA would have to be made up to fill in there, as that is definitely not how it happens. Here's a semi-conservative method. The original strand is going to be unzipped, and each of the two new strands contains one original strand, that dark blue, and one new strand, which is the complementary template for the other strand. This is how it happens. Now, on the next slide, we're going to see some good stuff. On this slide, this is a very good slide, a very informative slide here. Let's look at one thing first. Let's look at the far left, and you see that this dark blue double-strand helix has a 5' and a 3' end, right? They're labeled there. Let's look at the top one, the 5'. Go all the way to the other end of that molecule is 3'. And let's look at the 3'. Go all the way to the end of the molecule, and there's a 5'. That's what I was telling you. Each end has a 5' and a 3'. Now, in order for this to be copied, this thing is labeled, and this is pretty good. Number 1, helicases. Helicases are enzymes which untwist and unzip double-stranded helixes. So helicases unwind the parental double helix. That's these two little oval things here. So that's how the two strands got opened up or separated. It broke the hydrogen bonds and separates the strands. Now they want to go back together, but look at number 2. Number 2 stops them. Single-strand binding proteins stabilize the unwound parental DNA. It will not let the two strands go back together yet. So it keeps them apart. And where it's keeping them apart is called the replication fork, where the two strands of DNA are unzipped. That's the replication fork. Let's look at number 3. The leading strand is synthesized continuously. That means there's no breaks. It flows from the start to the finish in the 5' to 3' direction by DNA polymerase. Both DNA and RNA are laid down in the 5' to 3' direction when they are copied. So look at this little light blue strand. You see that there's the 5' end, a new 5' end, and look, it goes all the way down goes to the left, and there is DNA polymerase. DNA polymerase, if you look at the way that that's worded, ASC is an enzyme. Read it backwards. An enzyme that makes a polymer of DNA. That's what DNA polymerase is, an enzyme that makes a polymer of DNA. So it locked onto the 3' end, and it starts laying down this new strand of DNA in the 5' to 3' direction, from the right to the left. So when it finishes, this will be the end of this new strand that it's making. The very end will be a 3' end. Now it says this one's called the leading strand because it's put together just smooth all the way down. There's no breaks. But look at the lower strand. The lower strand, which is the dark blue on the lower side, the lagging strand. Okay, lagging strand means that it's going to be a little bit slower, and it's actually put together in pieces. It says the lagging strand is synthesized discontinuously. That means in pieces. New sentence. Primase, an enzyme that lays down a primer, is what that means, synthesizes a short RNA primer, not DNA. That little red thing there that is pointing to is RNA primer. which is extended by DNA polymerase to form an Okazaki fragment. Some guy named Okazaki, or Mr. Okazaki from Japan, got his name put on this one when he discovered this. So primase is going to lay down that little red piece of RNA on the DNA. Now that gives a base for DNA polymerase to lock onto to lay down the new light blue colored DNA in the 5' to 3' end direction. Okay, so you can see that as you follow the arrows over to the right, it's going to be a new 3' end. The problem is that DNA polymerase cannot join pieces of DNA together. So when the DNA polymerase butts into the primase, the primer, it knocks the primer off, but it can't join the two fragments together. You see that they're on pieces. Look at number 5. After the RNA primer is replaced by DNA, so it's knocked off and DNA will replace it. Keep reading. DNA ligase joins the Okazaki fragments to the growing strand. Ligase can fill in the missing bases. And so it'll join the far right one, which we'll call like number 1, to number 2, the next one to the left. It'll join that one to the next one to the left, and so on. And so it's going to put the pieces together going from the right to the left until that strand is made. And then you'll have 2 strands of DNA. And when we talked about chromosomes copying themselves, these are the 2 sister chromatids that are being made, and they'll be held together in the middle, remember in the centromere, by a kinetochore. DNA repair. We talk about DNA proofreading. There are many enzymatic pathways which check DNA. It says proofreading. In bacteria, errors are corrected by DNA polymerase as it checks for accuracy. In eukaryotes, since we have many chromosomes and they're linear, DNA polymerase and additional proteins check for accuracy. So we have a lot of proteins that are checking to make sure our DNA code is good and there's no problems with it. More than 50 different DNA repair enzymes are involved in excision repair. They can pull out mutations and repair those spots, put in the correct sequence of bases. Either directly repairs the mistake or the damaged segment is excised, cut out by one repair enzyme, and the gap is filled by the activity of DNA polymerase to lay the bases down and DNA ligase to tie the 2 strands back together. Now there's something called telomeres. Telomeres are on the end of a chromosome, and that's what DNA polymerase is going to lock onto when it copies the DNA. That's where it starts, on the end of the chromosome that has the telomeres. Now we're not going to hold you for this sequence here, but just know that DNA polymerase is going to lock onto the telomere, and then it's going to copy the chromosome. The problem is that it doesn't copy the part that it's laying down on, that it attaches with, or attaches to the chromosome. So every time the chromosome is copied, the chromosome gets shorter. It's reduced on the leading strand. So the chromosome gets shorter every time the cell divides. And that's considered to be part of aging in humans and other life forms. Telomerase is an enzyme. Well, our telomerase gene is turned off in most of the cells in our body. But in cancer cells, telomerase is turned back on. Cancer cells in the germ cell lines, the ones that make like sperm, are turned on. And so those cells don't age. Every time the DNA polymerase locks on and copies the DNA, telomerase comes back on and replaces the telomere. So the telomere never gets shorter. So cancer can live forever. There was a lady back in 1951 that had cervical cancer. Her name was Henrietta Lacks, and the cells they harvested from her cervical cancer actually grew into the first human cancer cell line. And they still have those cells today. They're called HeLa cells, named after her, Henrietta Lacks, and research has been done on those since 1951.